Welcome, my name is Agata Błaszczyk. This is a tutorial video describing the procedure of enzymatic assays. Trypsin inhibition will serve as an example. Small organic molecules produced by marine organisms are a vast source of novel bioactive compounds and potential drugs. One of the first steps in marine drug discovery is production of crude extracts from a selected marine source. Extracts containing bioactive compounds could be identified by different screening assays. Among natural compounds, enzyme inhibitors have been considered useful tools mainly due to their biotechnological potential in pharmacology and agriculture. In particular, protease inhibitors represent important examples of enzyme inhibitors. They are capable of inactivating the target proteases in the presence of human diseases. For example, in arthritis, muscular dystrophy, pancreatitis, thrombosis, different cancer types, and AIDS. We shall start from studying the mechanism of enzymatic reaction. Enzymes are usually proteins that possess one or more active sites ready to bind a substrate. During the enzymatic reaction, substrate binds to enzyme catalytic center. Enzyme-substrate complex is formed. When the reaction is finalized and the created product is disconnected from the catalytic center, the enzyme returns to its original shape and is ready to bind another substrate. However, some disturbances might occur. There are many types of molecules that are capable of interfering with the enzyme's activity. These are called inhibitors. We can distinguish two types of inhibition: reversible and irreversible. Reversible inhibition can proceed mainly in two ways. Competitive inhibition occurs when substrate and inhibitor compete for one and the same catalytic center. The second type of reversible inhibition is a non-competitive one. It takes place when substrate and inhibitor bind with the enzyme. However, the formed complex is catalytically inactive. Crude extracts, isolated fractions, or pure compounds can be tested in this assay. What's important, the samples have to be transparent, colorless, and free of any dirt. And remember, your samples in their final form have to be dissolved in water only. To properly conduct an enzymatic assay, you should have access to appropriate laboratory equipment: automatic pipette, multi-channel pipette, microplates with covers, plastic reservoir used for the substrate distribution, and full ice container. In addition, you would need a plate reader with adjustable temperature that would allow you to make measurements at 405 nanometers. Which reagents should be prepared in advance? You would need buffer, trypsin solution, a protein in solutions in the given concentration range, and substrate solution. Procedures for preparing reagents and their concentrations are given in the table. Remember to prepare the substrate solution on the day of conducting the assay. Do not expose it to the light. The next step is to design the plate. If you do it properly, reading the results would be much easier. Place the negative control, blue color, in the wells A1 and B1. The wells from C1 to H2, yellow color, should be filled with increasing concentration of our inhibitor, each in 2 replicates. The gray wells are intended for your 8 samples, each in 3 replicates. And remember to apply the samples in an increasing order, from the lowest concentration to the highest. The table shows the order in which reagents are applied to the plate. Please have a look how I fill the plate. In the first step, apply 10 microliters of water to the negative control wells and zero concentration inhibitor. Then apply 10 microliters of different inhibitor concentrations to C1, H2 wells. Then add 10 microliters of your samples to the remaining wells according to the previous plate design. The next step is to fill the wells with 10 microliters of our enzyme. For convenience, I rotate the plate and apply the enzyme from the other side. Why? Just to avoid early mixing of enzyme and inhibitor. Remember that the enzyme solution should be kept on ice throughout the test. When the enzyme is finally added to all the wells, the plate should be gently mixed so as to connect all the reagents without splashing them. Next step would be adding 100 microliters of the buffer to each well. Pipetting is facilitated and accelerated by using multi-channel pipette. Afterwards, cover the plate, mix and incubate it in 25 degrees for 5 minutes. When this time passes, add 100 microliters of the substrate to each well. Again mix the plate and incubate it in 25 degrees. Measure the absorbance after 10 and 20 minutes. These are the results of our test. The wells with negative control remain colorless, whilst the ones containing the inhibitor are yellow. What's important, the color is inversely proportional to inhibitor's concentration. It means that lower concentration gives more intensive color. Next, on the basis of the measured absorbance, a standard curve for the inhibitor should be drawn. Your role is to compare the obtained results with the curve. Calculate the IC50 with the use of regression method. Of course, the inhibition assays for other enzymes from the protease group can be carried out in analogous manner. Thank you for your attention.